Hi, everyone. So we're going to continue with chapter 10. This is continuing with microbial metabolism. This is part 3, catabolism, getting materials and energy. So this is all about the breaking down reactions, and the overall reactions are depicted here in this screen, where we start with glycolysis with glucose as the substrate. As we go through all the steps of glycolysis, we end up with 2 pyruvates, and we get a couple of ATPs out of the deal and some NADHs. And then we go into the prep cycle for transition reaction into the Krebs cycle, where again, multiple reactions occur, more NADHs, some FADH2s, and some ATPs as we continue to oxidize the pyruvates. Then all of these NADHs and FADH2s go into the electron transport chain and through chemiosmosis, and we generate lots and lots and lots of ATPs. And if we're using oxygen as the final electron acceptor, which we do in aerobic respiration, we can end up with water, carbon dioxide as waste products, and we can end up with a total of 38 ATP. So let's go through this whole process. So this is the catabolic processes. And remember, catabolic is breaking down. It's a hydrolysis reaction. Remember, you're using water to break those bonds. Energy releasing and exergonic. So all of those terms are used to describe catabolic reactions. So these are the learning outcomes from this section. You see that there's quite a few of them, and we're going to go through these steps fairly quickly, but still in a little bit of detail. So keep that in mind. This video will be a little bit longer maybe than some of the other videos. So when we look at catabolic reactions, if you remember from the graphic that we saw earlier, this is the area that we're going to be looking at. The last section of this chapter that we're going to talk about are the anabolic components, where we can use some of these metabolites to build the molecules of life. We will not be covering photosynthesis, which is section 10.5. So just ignore that. You don't have to even read it. But of course, if you're interested in photosynthesis, feel free to, but you won't be examined on it. So the 3 basic pathways that we're going to talk about here are aerobic respiration, anaerobic respiration, fermentation, and of course, glycolysis. So these are the 3 main. Glycolysis is the most common pathway that's used to break down glucose. Pretty much everybody does it. And from there, cells can go, or they can go on and either do fermentation, anaerobic respiration, or aerobic respiration. So let's look at these 3 main pathways. So if we look at the 3 main pathways, we can see in the left-hand panel, this is glycolysis. Notice everybody's starting with glycolysis. This first one is the one we'll talk about for the most part, is aerobic respiration. And this is where oxygen is being used as the final electron acceptor and is being used to generate lots and lots of ATP. We'll go through this whole process in a bit more detail in the coming slides. We can also, well, we can't, but some bacteria can do what we call anaerobic respiration. And here, instead of oxygen as the final electron acceptor, they use other compounds like sulfates, nitrates, those kinds of things as the final electron acceptor, and can also generate ATP. Notice the rest of these processes are the same. It's just where it's different is what is being used as the final electron acceptor. And this is important because some bacteria can use this anaerobic pathway to generate tremendous amounts of ATP and not have to use oxygen. As we've mentioned before, oxygen can be toxic to anaerobic cells. So this is one mechanism that they can do. Now, some cells can't do these 2 processes of respiration at all. So instead, they will do fermentation. So first, they'll start with glycolysis, and then they'll undergo fermentation. The products of fermentation are either going to be alcohols, acids, mixed acids, and we'll talk about some of those as we go on. Even our cells can do fermentation. So if we get to a situation where we're exercising and our muscles just we can't keep up with the oxygen flow, you're breathing heavy, but you just can't get enough oxygen to your cells, we will do fermentation. And humans, of course, when we undergo fermentation, we can only make lactic acid. But we'll see other products are possible if you're a microorganism. So this is comparing aerobic and anaerobic respiration. Both of them are converting glucose to carbon dioxide. So they're both oxidizing glucose all the way down to carbon dioxide. The final electron acceptors are where it varies. They both use the same pathways, and the yield is just slightly lower for anaerobic respiration as compared to aerobic respiration. So let's take a closer look at aerobic respiration. This is where oxygen is the final electron acceptor. We're starting with glucose, and we carry out glycolysis, the Krebs cycle, and then the electron transport chain, or the respiratory chain, for oxidative phosphorylation and chemiosmosis. So we're going to go through each of these processes and what the steps are. So we start with glucose, and just to remind you, this is a glucose molecule. It's a 6-carbon sugar. It's called a hexose sugar, hexose for 6 carbons. And you can count the carbons: 1, 2, 3, 4, 5, 6. Each of these corners represents a carbon. And notice again, just to remind you, carbon forms 4 bonds. So each of these will have 4 bonds. Not shown will be an oxygen, if for the additional bond, just to make it a little cleaner to look at. And glucose is readily oxidized. It readily gives up and loses its electrons and hydrogens to where it ultimately will become CO2. Other molecules can also enter in this pathway. The isomers of glucose, remember, these isomers have the same molecular formula, so both fructose, galactose, also are made up of C6H12O6. However, the organization and the structure of those atoms are a little bit different, and that's why they're isomers. And also some fatty acid subunits can also enter into these catabolic pathways, particularly the glycerol portion of it. So here's how it all starts, and this is for all 3 kinds of processes. Glycolysis, again, we see that whether you're doing aerobic respiration, anaerobic respiration, or fermentation. And so here, the whole process is to enzymatically, that means using enzymes to get to pyruvic acid. So pyruvic acid is 3-carbon sugar. So basically, this whole thing is just basically breaking glucose into 2 pieces. Is kind of what we're doing here. So this is usually the first phase of aerobic respiration. It's the primary metabolic pathway for a lot of cells, and pretty much everybody does glycolysis. It synthesizes a small amount of ATP anaerobically. You only get 2 molecules of ATP net, and we'll see that in just a moment. And we get pyruvic acid, which is a central mediator. So this is showing the entire process of glycolysis. We're not going to worry about all of the different transformations that occur. We are going to watch a couple videos. They're a little bit long, and they will make this talk a little bit longer. You can skip through them and watch them later on your own time, or you can watch them embedded in this video, whichever is better for you time-wise. But I strongly encourage that you watch them. I spend time curating and finding videos, and I usually try to find ones that I think are going to give you good information. So we can see we're starting with glucose, and we have to put in some ATPs at the beginning. This is called priming priming the reaction and getting the reaction started. So we first add a phosphate group and we end up with glucose 6-phosphate. Then we have to add a second phosphate, so we can end up with and we have an isomerization to convert it to fructose 6-phosphate, and then we get to 1,6 fructose 1,6 diphosphate. So notice there's phosphates on either side. That's where the ATP came into play. And then we can break that in half and split this molecule into 2 3-carbon sugars. We have to go through some isomerizations and some conversions and so on and so forth. We end up adding hydrogens and electrons to NAD to NADH, so that can carry those electrons over to the electron transport chain. And we can see we generate a couple ATPs through this process. If we were to add up all the ATPs that we get in this process, we would find that we get 4 from one glucose molecule. You see 1, 2, 3, 4. However, it cost us 2 at the beginning. So our net gain is 2 ATPs from glycolysis. And we also have NADHs being made. We get 2 NADHs, which of course they're at higher energy as well, and we get 2 pyruvates. So let's watch this process. Here again, the input's 2 ATPs plus glucose plus 2 NADs. The outputs are 2 pyruvates plus 4 ATPs, net gain of 2 ATPs and 2 NADHs. Let's watch this video. This is showing us what the enzymes look like, and we'll watch this whole process. Now, again, you don't need to memorize all of the intermediates or the enzymes that are being used, but I do think it's helpful for you to kind of get an idea and get a visualization of what these things look like to make it better and easier to understand. So here we go.
The sweet, energetic glucose molecule is the energy source providing power for your living cells. Glucose from the food you eat is slowly broken down into 2 pyruvate molecules, extracting energy in a sequence of enzyme reactions called glycolysis. The glycolysis pathway requires energy to begin, delivered by chemical fuel ATP. The primary source of ATP in your cells are mitochondria organelles. Deep inside mitochondria, chemical reactions of aerobic respiration consume oxygen from the air you breathe to generate vast amounts of ATP. Sitting on this abundant supply of ATP is the first enzyme of the glycolysis pathway. The reaction of the first enzyme transfers a phosphate group from ATP to glucose, creating glucose 6-phosphate. The second enzyme rearranges glucose 6-phosphate into its isomer, fructose 6-phosphate. The third enzyme uses a second molecule of ATP, creating fructose 1,6-bisphosphate. The 6-carbon sugar is now ready to be broken apart. The fourth enzyme cuts the molecule in half, creating two 3-carbon sugars, each with a single phosphate attached. The sugar products are isomers of each other, but only glyceraldehyde 3-phosphate is ready to continue with glycolysis. The sugar isomer, dihydroxyacetone phosphate, must first be converted through isomerization before continuing to the sixth enzyme. The sixth enzyme adds a second phosphate to glyceraldehyde 3-phosphate, creating 1,3-bisphosphoglycerate, while 2 electrons are transferred to NAD+, which is reduced to NADH. Halfway through glycolysis, the cell has consumed 2 ATP molecules, breaking down glucose, and is now ready to capture energy in return. The seventh enzyme transfers a phosphate group from 1,3-bisphosphoglycerate to ADP, creating chemical fuel, ATP. The eighth enzyme rearranges the phosphate from the end of the molecule to the middle. The ninth enzyme catalyzes dehydration, increasing the potential energy in preparation for the final glycolysis reaction. The 10th enzyme transfers the remaining phosphate group to ADP, producing ATP and the 3-carbon sugar, pyruvate. Glycolysis finally produces a net gain in ATP. The glycolysis pathway breaks down glucose into 2 molecules of pyruvate, gaining 2 ATP and 2 NADH in the process.
Okay, so I like this video because it gives us kind of a real sense of sort of what the enzymes look like and how they're behaving inside the cell. And keep in mind, I mean, they show it kind of crowded and densely packed. And if you keep in mind that these enzymes are carrying out these reactions up to millions of times per second, you can just imagine how fast this is all happening. And it makes sense when you think about how much ATP cells actually need in order to function. So pyruvic acid is the center of what we call a central metabolite. It's utilized by many different pathways in many different organisms. So we can see here in this graphic, pyruvic acid is in the center. After coming through glycolysis, that's the product. It can be taken then and used to make amino acids, sugars, and fat metabolites. So it's a building block of these components that are needed to build cells. It can also be used in fermentation to produce a number of different products there if oxygen is not available, or it can continue to be oxidized and carried through cellular respiration, aerobic respiration, whether well, aerobic or anaerobic respiration. Right now we're just talking about aerobic respiration. So the next step to that is the Krebs cycle, or it's also called the citric acid cycle. And so you'll see it written as TCA, the citric acid cycle, or the Krebs cycle. It's named for the person that discovered it. So in order to enter into the pyruvate acid to enter into the Krebs cycle, it first has to go through a conversion, and it gets converted into acetyl coenzyme A. And that's important for it to have, that's an important first step. This oxidation reaction releases the first carbon dioxide molecule, and that's this as oxidized as it can get, as CO2 can get. There's a cluster of enzymes, and coenzyme A dehydrogenate pyruvate acid into a 2-carbon acetyl group. NAD gets reduced to NADH, and NADH formed, again, is shuttled to the electron transport chain, which we'll talk about later. So that's in the transition reaction. So once that occurs, then it can enter into the Krebs cycle, or the citric acid cycle. And here we can see an overview of this cycle as well, with all the various steps, where we get NADHs and FADH2s, and where we get ATP directly through this substrate level phosphorylation. So we're going to watch a video on this process as well. Keep in mind that all of the reactions of the Krebs cycle are going to happen twice, because we have 2 pyruvates. This is from each glucose molecule. So just kind of keep that in the back of your mind, that this cycle is going 2 times for every glucose molecule. So you can double the items. Instead of 1 ATP, or you can say 2 ATPs, because the first pyruvate went through, and then the second pyruvate goes through. There are several, it serves as a way to transfer the energy from the acetyl-CoA and NAD pluses and FADH or FAD by reducing them. So the NAD gets reduced to NADH, and the FAD gets reduced to FADH2. And so we end up with NADH, FADH2, as well as ATPs that are produced directly. So again, this is just a closer look at the Krebs cycle. One of my first jobs was actually looking at one of the enzymes here. I worked with isocitrate dehydrogenase. But even that being the case, I still don't have all of the steps and all of the enzymes memorized. And I certainly would never expect you to do that, to memorize those as well. Perhaps if you go on and take advanced biochemistry classes, you may have to. At one point in my life, I had these things stuffed into my brain, but there's no reason to memorize all these enzymes. But if you know what goes in and what comes out, that's the key here. So we're going to go ahead and watch this other video on the Krebs cycle. Now, I want you to note as you're watching this that they're showing for a eukaryotic cell. So they're going to talk about how this is happening in the mitochondria. Bacteria don't have mitochondria, so the Krebs cycle actually just happens out in the cytoplasm. The electron transport chain, which normally happens in the cristae of the mitochondria in eukaryotic cells, will actually happen in the cell membrane of the prokaryotic cell, or the bacterial cell. So just keep that in mind, that the location is going to be different for bacteria, because bacteria don't have mitochondria, but the actual cycle itself is very similar. So let's take a look at this video. Again, this is a fairly long video with lots and lots of detail, but I think it's useful to really kind of understand what's happening in the citric acid cycle, or Krebs cycle.
The origin of energy production and biosynthesis inside your living cells is the dynamic mitochondria organelle. Mitochondria are filled with metabolic enzymes that catalyze a loop of chemical reactions called the citric acid cycle. The 8 steps of the citric acid cycle gradually break apart two carbon atoms from glycolysis, capturing liberated electrons for the electron transport chain, while generating carbon dioxide as waste. The citric acid cycle starts with the end product of a previous cycle, the 4-carbon molecule oxaloacetate. New carbon enters from glycolysis in the form of acetyl-CoA, a 2-carbon acetyl group attached to coenzyme A. Step 1 is the catalytic transfer of a 2-carbon acetyl group from coenzyme A to 4-carbon oxaloacetate, creating 6-carbon citric acid, the molecule that gives the cycle its name. Citric acid is used by your cells for the biosynthesis of fatty acids, lipids, and cholesterol, and is also the substrate for step 2 of the citric acid cycle. The enzyme reaction of step 2 makes a small change to the citric acid molecule, moving the position of an oxygen atom, converting citrate into isocitrate. Step 3 of the cycle removes a carbon atom, forming carbon dioxide as waste, while converting 6-carbon isocitrate to 5-carbon ketoglutarate. During this process, chemical energy is harvested when 2 electrons are transferred to coenzyme NADH, which delivers them to nearby enzymes of the electron transport chain. Step 4 of the citric acid cycle is performed by a huge multi-enzyme complex, connecting multiple chemical reactions with flexible tethers, efficiently moving reactants between active sites, and diverting electrons between pathways. The large complex is made with a repeating unit of 3 core enzymes, working in sequence to remove a carbon atom from ketoglutarate, generating carbon dioxide waste, then attaching 4-carbon succinyl to coenzyme A. The electron from ketoglutarate oxidation is retained by the tethered enzyme before passing it to coenzyme NADH, which delivers electrons to the electron transport chain. Enzymes of step 5 separate succinyl from coenzyme A by breaking an energetic bond, providing enough energy to create GTP. GTP is a building block of RNA and an energy source for protein synthesis. In the most direct path to ATP from the citric acid cycle, other enzymes in the matrix can use GTP to make the chemical fuel ATP. Step 6 is performed by an enzyme embedded in the inner mitochondrial membrane. This enzyme is also known as complex 2 of the electron transport chain. The enzyme catalyzes the oxidation of succinate to create fumarate, releasing 2 electrons that hop through the interior to coenzyme Q, which is reduced. Coenzyme Q travels in the membrane, carrying the electrons to nearby enzymes of the electron transport chain. Step 7 takes fumarate, generated by step 6, and reacts it with water to create 4-carbon malate. The final step of the citric acid cycle uses malate to regenerate oxaloacetate, transferring electrons to coenzyme NADH, which supplies the electron transport chain. Oxaloacetate can return to step 1 for another loop of the cycle, and is also an essential building block for making amino acids and the genetic code of DNA and RNA. At the center of cellular metabolism, the citric acid cycle generates both chemical energy and material for biosynthesis of the entire diversity of molecules found inside a living cell.
You'll see it called both. I generally refer to it as the electron transport chain or electron transport system, but you'll see it in both ways. So here, this is something that occurs in the mitochondria. Typically, if you're a eukaryotic organism and you have all of these proteins embedded in the inner membrane of the mitochondria, in the cristae, if you recall correctly, from your earlier biology class. So if we were looking at mitochondria, here's our cell, and well, let me make a bigger cell. Here's our cell, and here's our mitochondria with that inner membrane, and those proteins would be embedded in this inner membrane here. But we're talking about bacteria, so where is that going to happen? In bacteria, where that happens is in the plasma membrane. So this is looking at a gram-positive cell, but it's true for a gram-negative cell as well. You've got the plasma membrane with the phospholipid bilayer. Remember all the various proteins and things that are embedded in the phospholipid bilayer, including these proteins that are needed for the electron transport chain. And this is why if you disrupt this, as we'll see later in the semester, we talk about how to get rid of bacteria, how to control the growth of bacteria, can enter into the Krebs cycle at various points along the way, depending on what groups it is. So that brings us to the end of this topic, and I understand this has been a long one. So feel free to kind of fast forward through the videos or to just go back and look at little bits and pieces, and have a good day.